So what we're doing today is we've got the fundamentals of protein structure and macromolecular structure out of the way. We talked about sizes and shapes. We did shapes of proteins, and they're mostly globular, but elongated ones as well. And you also get, I think David Scott will be talking about this in the structural biology module in the new year for ABTs and ABIs. If you choose that module, you've got a choice between that and the genetic analysis and bioinformatics module. But yeah, David will be talking about, I think I'm sure he'll mention intrinsically disordered proteins. But generally speaking, they're globular with some exceptions. And polysaccharides, as we saw yesterday, have a much greater variety of shapes. So we've given an introduction to proteins and polysaccharides and glycoproteins. The degree of glycosylation represents the amount of sugar on the molecule. But we're focusing on protein structure today, and we're going to show you the Rasmol software, which is already on the Moodle ready for download. But before that, we're going to show two videos. It's an easy way of teaching, actually. If you go into teaching later on, you learn this. It's called getting a balance of teaching material. But there's two excellent videos by my former mentor, Sir David Phillips, who gave the lysozyme, well, not the lysozyme, the little protein folding clip we saw, I think 2 weeks ago now, was it? Or maybe 2 weeks, 1 week ago. Yeah. So there's two other clips from him. One is hemoglobin, and I want to show you that because it's not just about hemoglobin and how subunits come together when the thermodynamics is favorable, that is delta G is negative. So the monomers come together to form this 4-unit structure. But what happens when things change and there's a mutation in an amino acid or more than one amino acid? Now, in most cases, these changes, mutations are redundant, and they give species variation between different molecules, different hemoglobins in this case. And the mutation just has no major consequence. But sometimes it does. And there's an example, it's called Sickle-Cell Anemia, how just one change in amino acid can have a dramatic effect. But that's relevant to biomolecular technologists because point mutations, changing proteins or changing molecules for specific functions is, if you like, a biomolecular technological skill or tool. And sometimes it has an effect, sometimes it doesn't. But this is a natural thing, sickle cell anemia. And understanding it and these sort of similar processes helps us to understand how we can treat these things maybe in the future to eliminate these sorts of diseases. So the hemoglobin is very relevant. And then I'm going to show you the lysozyme because Phillips was the man, or he led the team which worked out the crystal structure of lysozyme. It was the first enzyme whose structure was determined. Before that, insulin with Dorothy Hodgkin, a small peptide. Then there was myoglobin with John Kendrew, and then hemoglobin with Max Perutz. These were guys that were on the DNA film, Kendrew and Perutz. Not to actors playing them, not the real Kendrew and Perutz. But lysozyme was the first enzyme that was solved by David Phillips. And I want to show you this because it shows you how crystal structures are developed. Again, in structural biology, we'll have Peter Moody from the University of Leicester coming up. He'll be talking about that. But showing how a crystal structure is, you've got to crystallize a protein first of all, but if it's crystallized, then how you get a structure out of it. But it's the enthusiasm, the way he presents this. This is his baby, you know. And that just comes across, I think, fantastically. So we'll show the lysozyme, and then we'll look at lysozyme using Rasmol. We'll look at our favorite protein, which is of albumin, which is not a protein, it's a glycoprotein, using Rasmol, and look at other things as well. It's really useful. And then when you're doing your, I haven't set you the work yet, have I? The assess work for this module. But there's, I think, an essay and something else where you can use Rasmol because you can get the molecule in the particular orientation you want to show alpha helices or something, or active site, and you can do a snapshot, JPEG. Then it's your picture in your piece of work, so you're not having to worry about quotations correctly and things, because it's yours. You know, you just see, you say, obviously you give accreditation to Rasmol, the software used, but it's your picture. So yeah, Rasmol is good. So let's start the video, and we'll have some curtains last time. You may have noticed last week when we had it's great having the film in the Chalmers room, but there was a problem with the lights. So you saw me messing about, and then various engineers came in, and we just couldn't get those lights at the back to switch off, I'm afraid. But I think it didn't disturb the view. It was reasonably okay. Is it worth recording a recording? Should we leave it running? Right, so it's hemoglobin structure. Now, let's make sure we've got the sound up. It's great, this modern technology. You know, Moodle is so fantastic. As academics, we all grumbled about it because it's something else new we had to learn, and there's a resistance to change. And but it was definitely worthwhile. I think it's a remarkable teaching tool. Okay, so what we after? Hemoglobin. This is about 10 minutes, 12 minutes, but it's very effective. And again, what students have done is they've actually played the video for themselves and then done a screen dump, because I think there's a question on what happens at a point mutation or something, the consequences, and they use the hemoglobin example, just do a snapshot. So yeah, use the video material as well for your work. Right, let's see if we can get full screen. Right, so let's see. Good. There's nothing in the middle, which is that full screen? Is that okay? Yeah. Sound is nothing. Oh, great. So what's happened there?
Molecules associated. Nanozymes as we turn to consider how protein molecules associate to form larger assemblies, it's illuminating to consider hemoglobin, the protein in the blood which transports oxygen from the lungs to the tissues. This is a multimeric protein made up of subunits of two different kinds. Here we see one of the alpha subunits in a space-filling form, which associates with one of the beta subunits, shown here in yellow, to form a quite strongly associated alpha-beta pair, related by an approximate twofold axis of symmetry. This pair associates with another identical pair to form the tetrameric molecule, which you see here. This molecule has quite high symmetry. There are two approximate axes of symmetry relating alpha and beta subunits, and there is a real axis of symmetry shown at right angles to this diagram, which relates the two alpha subunits and the two beta subunits. Let's look now in a little more detail at the contact between the alpha and beta subunits making up this alpha-beta pair. We turn the molecule first, we can look edge on at the contact region between the alpha and beta subunits, and if we now take away the space-filling representation, we can see underneath the skeleton of the polypeptide chain, which shows that the chain in this molecule is folded very largely in alpha helical regions. If we concentrate on the white and yellow subunits, notice that the regions that come together are related by zero twofold symmetry at similar regions, and the reason why they come together is that the subunits here have sticky patches made up largely of non-polar hydrophobic residues. The principles involved are just the same as those involved in the folding of the lysozyme chain, which led to a hydrophobic pocket in the middle of the molecule. Here, there is a hydrophobic pocket between two subunits. In addition to these non-polar contacts, there are also polar contacts involved, in which hydrogen bonds are formed between the nitrogen atoms, coloured blue, and the red oxygen atoms, but these are buried within a very largely hydrophobic region. So they form a strong contribution in which the partial electrical charges are compensated. Overall, the impression is of a mainly non-polar contact in which more than 125 atoms are involved. And this association is quite a strong one. This alpha subunit and beta subunit remain connected in this way throughout the activity of the hemoglobin molecule. Hydrophobic cores and sticky patches then are very important in aggregation of protein molecules. Let's look now at what happens if we turn to an abnormal hemoglobin which undergoes a further level of aggregation. Hemoglobin is involved in carrying oxygen in the red blood cells, and the red blood cells shown here have this nice characteristic disc-like shape, a biconcave disc. In sickle-cell anemia, however, the blood cells are sometimes distorted into these grotesque sickle shapes. And if we disrupt one of these sickle cells, we find within it a mass of fibers formed, rather than a concentrated solution of hemoglobin. The next stage in our investigation of the aggregation of protein molecules, then, is to consider how it is that hemoglobin molecules can come together to form these fibers in sickle cell hemoglobin. If we look at normal hemoglobin, the tetrameric molecule, in a space-filling representation, we see that the surface, as usual, is largely polar in character, with nitrogen and oxygen atoms exposed upon it, but together with a number of non-polar residues here and there. If we take away the space-filling atoms, apart from one or two, to expose the skeleton, we can concentrate on this residue, which is a glutaric acid residue, number 6 in the beta chain. In sickle cell hemoglobin, this polar residue is changed to a valine residue. That's a change to a totally non-polar residue exposed, as we see, on the surface of the molecule. Elsewhere, on this right-hand side of the tetramer, there is another pocket down here, which, despite the appearance of some oxygen atoms in it, is largely non-polar, made up of a phenylalanine and a leucine side chain, which are exposed only in the deoxygenated form of hemoglobin. That's hemoglobin which is not carrying oxygen. In other places on the molecule, there are also some small sticky patches, in particular in this view at top and bottom, where we see them again including some polar atoms, but largely non-polar in character. But these sticky patches are not very extensive. They're not large enough to lead normally to the aggregation of hemoglobin. But they are large enough to lead to the transient formation of aggregates involving 2 or more hemoglobin molecules, and we can see such a dimer now in which 2 of these sticky patches, the top of one molecule and the bottom of the other, have come together. But this is a rather weak interaction, wouldn't persist for very long or lead to the formation of even larger aggregates unless it were reinforced in some way. Now, in sickle cell hemoglobin, we can imagine another such pair of molecules approaching our first pair in such a way that the new valine side chain forms a contact with this pocket which involved phenylalanine and leucine. And similarly, the phenylalanine-leucine pocket on our new hemoglobin forms a contact with the valine on the bottom hemoglobin of our first pair. Now this means that this molecule is linking together these 2 molecules and reinforcing the effect of this rather weak sticky patch, enough to make this a rather permanent aggregate, which, as we see, links together these 4 molecules. And since this contact is exposed now down here, and this contact is exposed up here, this is an open-ended form of aggregation, so that if we look at this tetramer, and then how it might build up into a polymer in a slightly less well-resolved picture, we can see that long chains of hemoglobin molecules might form. We replace these by red representations for each tetrameric molecule. We now have a long fiber with helices of hemoglobin molecules intertwined. That seems to be the way in which sickle cell hemoglobin molecules aggregate to form the fundamental fibers. And if we go back to the electron micrograph showing one of these fibers in detail, we can then begin to see whether this structure is observed. We home in on this picture, looking at it at slightly larger magnification, then we can begin to see regions such as that one, where one can just about discern the separate hemoglobin molecules forming this long, twisted helical array. So in this way, we can understand how it is that a simple change in the hemoglobin molecule, a change of 1 amino acid residue in each of the beta subunits, can lead to the formation of long, fibrous arrays of hemoglobin molecules that distort the shape of the red blood cell into the shape that's characteristic of sickle cell anemia. And what's more, we can go on and try to intervene in this molecular situation in such a way as to ameliorate its pathological effects.
Okay, thank you, Sir David. What happens, you mentioned that this was deoxyhemoglobin, but what happens in oxyhemoglobin? Yeah, so this sickle cell case only happens in deoxyhemoglobin, and the reason is that in oxyhemoglobin, when oxygen's bound, there's a change in conformation of the beta subunit, and we had that mutation of a glutamic acid going to a valine, giving an extra sticky patch. But there's no sticky patch. There's also those weak sticky patches at top and bottom, but also at the bottom on the side was that hydrophobic pocket of phenylalanine and leucine. And he said that that only exposes itself at the surface for the deoxyhemoglobin case. So when you get oxyhemoglobin, that is tucked away from the surface and the whole complex breaks down. So it only applies to oxyhemoglobin. And it's really bad luck that that mutation happened, that glutamic acid to valine. And bad luck that those other interactions, which otherwise would be very, very weak, happen to get reinforced by the new hydrophobic interactions which were introduced. So I think that's an excellent demonstration of the effects of a mutation and how molecules interact, weak interactions and strong interactions. He mentioned hydrogen bonding and he mentioned hydrophobic bonding between non-polar molecules. So hemoglobin's a great example. And he also said that in the normal state, hemoglobin exists as tetramers, again through strong hydrophobic interactions. As you decrease the concentration, lower and lower and lower and lower and lower, then the hemoglobin molecules, the individual monomers will come apart, but not at the concentrations found in the body. You tend to find that things get more aggregated or associate as you increase the concentration as opposed to decreasing the concentration. And it relates to a law called Le Chatelier's law, what we did in chemistry at school, Chatelier's. And that is if a system changes, or if an environment, the system by itself changes, then the system will react to lessen that change. So if you reduce the concentration, you're lowering the numbers of molecules. So what does the molecule do? It splits into four to make more molecules. Yeah. So that's what happens. And it only happens with these non-covalently attached subunits. When you've got covalent bonds between subunits, then changing the concentration has no effect. But for associations, non-covalent associations like hemoglobin, and like our favorite milk protein, which is beta-lactoglobulin, that forms dimers and can form octamers, high concentration. So, but by lowering the concentration, you favour the monomer form. Okay, that's a good video. That was good. That, yeah. You can play it again yourselves because it's on the Moodle. Right, the second one, whoops, is a slightly younger David Phillips. This was recorded before this one was, but you can just sense the excitement. Just warn you that some of the things he's doing would not be allowed now by modern health and safety. Just wandering in near an X-ray goniometer while it's on and things isn't. You wouldn't be allowed to do that. But just to warn you, times have changed a little bit. Right. So, lysozyme structure. Come on. Waiting for Nottingham. Why are you so slow? Sorry, recording. University of Nottingham, very, very good. So quick. Right, come on. Okay, just see how problem solved. This is all about problem solving in science. What's this?
The walls of these bacterial cells are gradually being broken down by an enzyme, lysozyme. Molecules of lysozyme are in fact breaking more and more chemical bonds in the cell walls until the walls finally burst. That recording was taken through a microscope with a magnification of about 2000. So of course we couldn't see the individual lysozyme molecules in action. In order to see the detail in such a molecule, we would need at least 1,000 times greater resolving power than even the most powerful optical microscopes. But still, we didn't understand the activity of lysozyme until we had succeeded in looking at it in this detail. Lysozyme was the first enzyme whose structure was worked out, and the story of how that was achieved is the subject of this program. It begins with the chemical makeup of lysozyme. We knew that the molecule consists of a single polypeptide chain with 129 amino acid residues. Let's look at it again, starting this time at the amino end. And note, as we do so, that in these models, all the atoms other than hydrogen are represented by little balls, and the hydrogens are to be thought of at the ends of the rods coming out of them. The amino terminal residue is a lysine. A little further on, at position 6, we have a cysteine residue. There are 8 of those. A little further again, a cluster of alanine residues in positions 9, 10, 11. And so on. A little further along, we come to the second of the cysteine residues, 30, which we shall be meeting again later in the program, along with a nearby phenylalanine residue, 34, distinguished by its benzyl side chain. Next to it is a glutamic acid residue, 35, which is involved in the activity of the enzyme, and a little further along again, an aspartic acid residue, 52, which is also involved in the activity. And so it goes on through a rather random-looking arrangement of the residues. Sometimes with some standing out as these tryptophanes, 108 and 111, and so on to the carboxyl end of the chain, a leucine residue. So that's the chemical structure, and this is what we eventually determined to be the three-dimensional structure of the molecule. That long polypeptide chain is folded up into this compact globular shape, seen here in a model 100 million times larger than life. Now I know this must look rather a tangle, but by the end of the program I hope you'll have a much clearer idea of it, because you will need a clearer idea when we come in the next program to consider the activity of lysozyme, because the activity depends essentially on the three-dimensional structure. So let's get on with how we did determine this three-dimensional structure. The problem is that atoms in molecules are very close together, so that to see them in a microscope, we must use a radiation with a very short wavelength. Now, X-rays have the right sort of wavelength, and X-ray crystallography is the method, as you might know from S100 and the correspondence literature for this course. The first step, then, was to record the X-ray diffractions from a crystal of lysozyme like this one. The equipment we used was similar to this. X-rays are produced at the front here and emerge from this tube to impinge on the crystal here. The crystal typically contains something like 1,000 million molecules. The X-rays are diffracted by the crystal and finally end up at a detector unit which transmits the readings along here to a computer. This is done thousands of times with the crystal at different orientations. That is, between different measurements, the orientation of the crystal is adjusted by rotation about 3 different axes. For each individual measurement, the position of the detector unit also has to be adjusted. The appropriate angles between measurements are calculated by the computer. In the next step, the thousands of measurements are combined in a computer calculation to give maps of the electron densities like this. This map is a cross-section through the molecule, and the electron densities are shown by these contour lines, like the contours in a geographical altitude map. Such maps are calculated for many parallel cross-sections through the molecule, and they're drawn out on transparent sheets. So we get a three-dimensional picture of the electron densities in the molecule, where the electron densities correspond to atoms or groups of atoms in the molecule. It's difficult to see on this what I mean by densities corresponding to atoms. So here we've got another stack of cross-sections showing the simplest situation. In fact, these are the electron density contours for a single atom, and the center of the atom will be at the center of this ball of contours. So you can see how it should be possible to study a map like this and determine the positions of the atoms in it. But unfortunately, it's not quite so simple, because the resolution of the map isn't quite good enough to show the atoms separately, unless they're relatively far apart in van der Waals contact with one another. Instead, we have to recognize the shapes of particular groups and identify them with the residues in the sequence. But supposing that we've done that, how do we know which group we are looking at? This is where our knowledge of the sequence comes in. Here we have the amino acid sequence, drawn out again in a different way, folded on itself so that the cysteine residues come together in pairs to form disulfide bridges. There are 4 of these disulfide bridges, and since sulfur has a high electron density, features corresponding to these bridges should appear in the map as regions of high electron density. In fact, on a subset of 6 of these sheets, we can see such a feature, and I've sorted that out earlier for you to look at. Clearly, this feature here represents a group containing a patch of very high electron density. It is, in fact, the highest in the whole map. Now, say we interpret the features of the map having high electron density and shapes like this as the disulfide bridges. As we've seen, there are 4 of these, and we need to know which one we're looking at. In order to do that, we shall have to identify some other features in the neighbourhood. A good amino acid residue to look for now would be phenylalanine, since it has a distinctive benzyl side chain. Here's a set of 8 sections of the density map on which we can see the characteristic shape of phenylalanine. Now, if we look back at the sequence, we see that only one of the cysteines, 30, is exactly 4 residues away from a phenylalanine, 34. So we look in the map for the characteristic shape of phenylalanine separated by 4 residues from the dense contours of a cysteine. When we've done that, we shall know where we are. Now, the part of the map I just showed you with the dense contours in one corresponding to cysteine and the shape of phenylalanine in the other actually follow one another in the sequence, as you will see when we put them together. Here's the cysteine and here's the phenylalanine. It's rather difficult to see the number of residues between them unless you're actually personally handling the stack of maps. But we can make it a bit clearer if we take only a few cross-sections and gradually build up more cross-sections. Here's the cysteine residue. If we add another 2 cross-sections, you can see the residue next to it. If we add more cross-sections, you can see the next residue. And so on. Finally, we see the characteristic shape for phenylalanine, 4 residues away from cysteine. Since there is only one section of the chain when this happens, we know we've identified residues 30 to 34. We can see the sequence from 30 to 34 more clearly if we erase all the surrounding contours. In fact, this section of the molecule is part of a rather regular-looking section made up of residues 24 to 34. You will see this regular-looking section more clearly in a moment. In the meantime, you can see that once we'd located where we were, we could follow around the polypeptide chain in the electron density map, being encouraged to find characteristic shapes, for example, at tryptophan 63, and again at the cysteine, and so locating all the groups in the molecule. Then what we had to do was to transfer the positions from the map to build our model. That's how the structure was determined. But so far, I haven't really fulfilled my promise of explaining this tangle. And in order to do that, let's begin by looking at an alternative way of interpreting the map. This is a computerized system devised by Barry and North at Leeds University, and they display on the computer contours sectioned in 3 different directions, forming little cages of contours corresponding to the different residues. Here's the density map for residue 34, phenylalanine. Let's add the map for residue 33. We can now see how a representation of the molecule can be fitted to the map.
This is very clever.
Firstly, an orthogonal view of the map is displayed. Then, a representation of the molecule can be juggled about by a researcher who can translate the molecule and rotate it about its bonds until it fits the density map. The computer can now record the three-dimensional coordinates for this part of the molecule. We haven't time to go into more detail about this fitting process, but what we can do is use this type of display to show you the density map for residues 24 to 34. I said earlier that this looked like a regularly shaped section. If we rotate the map, you might see what I meant. There are a few gaps in the shape because we've chosen to display only the high electron densities, so as to avoid most of the side chains. But you may be able to detect the regular shape of a helix. The helical shape becomes obvious when we look at a representation of the molecule, omitting the side chains. In fact, there are three sections of helix in the lysozyme molecule, but instead of looking for them in this model, it's easier to see them without all the side chains. We've made a computer-animated film showing the molecule being assembled one amino acid residue at a time, so that you will see the structure building up. This isn't intended to represent the molecule being synthesized, but only to show these details of the structure. In the animation, most of the residues appear in a dark red color, but those in the helices appear in a lighter green color. Here's the first helix being built up. Actually, there's one more regular segment, a beta-pleated sheet, which starts at residue 41. So the molecule has 3 alpha helices and 1 beta-pleated sheet. You can see the overall shape of the main polypeptide chain more clearly when we've smoothed out the kinks in it, as we've done in this model. There's another distinctive feature of the molecule in its overall shape. At this angle, it's difficult to see this feature, but if we view it from above, we can see a distinct cleft in the globular shape.